So here's something worth thinking about as you settle in tonight. Right now, inside your body, there are roughly 30 trillion human cells. That's 30, followed by 12 zeros, an almost incomprehensible number. But here's the thing, there are even more viruses than that riding along with you, on your skin, in your gut, tucked into the lining of your lungs. Most of them aren't doing anything you'd notice. Some of them are actually helping, and a few of them, under the right circumstances, can bring the whole system to its knees. Viruses are everywhere, in numbers that make every other living thing on the planet look scarce by comparison. There are more viruses in a single liter of seawater than there are people who have ever lived. They're in the deep ocean, in boiling hot springs, in Antarctic ice, in the soil beneath your feet. And yet, for most of human history, nobody had the faintest idea they existed. That's where I want to start tonight. Not with the complicated molecular biology. We'll get there, and it's genuinely fascinating when we do. But with the long, strange story of how we even figured out that viruses were real. Because it took a surprisingly long time, and the path to understanding them is full of brilliant people, wrong turns, lucky accidents, and a few moments that genuinely changed the course of human civilization. So get comfortable. Let your breathing slow down. We're going to take this nice and easy. For thousands of years, people knew about infectious disease without having any real idea what caused it. The ancient Egyptians wrote about what sounds like polio. Smallpox scars have been found on mummies dating back to around 1100 BCE, including possibly the mummy of Ramesses V, who died around 1145 BCE. The disease left distinctive pockmarks on the skin, and they're still visible after 3,000 years. Imagine that, the physical signature of a virus, preserved in dried tissue across millennia, and nobody at the time had any framework for understanding what had actually happened inside that body. The dominant theory for most of history was miasma, bad air. The idea that disease came from foul-smelling vapors rising from swamps, rotting matter, or contaminated ground. It sounds quaint now, but it wasn't entirely stupid. Places with bad smells often did correlate with sickness, because the same conditions that produce bad smells, stagnant water, decomposing waste, also tend to harbor disease. The theory was wrong about the mechanism, but it accidentally pointed people in a useful direction sometimes. Quarantine practices, which date back to at least the 14th century during the Black Death, were partly based on miasma thinking, and they actually worked, even though the reasoning behind them was off. The real breakthrough in understanding infectious disease came in the second half of the 1800s, and it came through bacteria, not viruses. This is important because the discovery of viruses only makes sense as a story if you understand that scientists first had to figure out germs in general, and then they had to bump into something that didn't fit the germ framework. Viruses were discovered, essentially, as the thing that wasn't a bacterium. So let's talk about that world for a moment. By the 1860s and 1870s, Louis Pasteur in France and Robert Koch in Germany were building the case for germ theory, the idea that specific microorganisms cause specific diseases. Koch, in particular, was methodical about this. He developed a set of criteria, later called Koch's postulates, for proving that a particular microbe caused a particular disease. You had to find the organism in every case of the disease. You had to isolate it and grow it in pure culture. You had to show that the cultured organism caused the disease when introduced into a healthy host. And then you had to re-isolate the same organism from that newly sick host. It was rigorous, elegant, and it worked beautifully for diseases like anthrax, tuberculosis, and cholera. But here's where things start to get interesting for our story. By the late 1800s, scientists were running into diseases that seemed to be infectious. They clearly spread from one organism to another, but they couldn't find the bacterium responsible, no matter how hard they looked under their microscopes, no matter how many culture plates they prepared. Certain diseases refused to reveal their culprit, and one of the first people to seriously wrestle with this puzzle was a Russian botanist named Dmitry Ivanovsky. Ivanovsky was studying tobacco mosaic disease. Now, this might not sound very dramatic. It's a disease of tobacco plants, not people. But it was economically important, and more crucially, it turned out to be the doorway into an entirely new understanding of life itself. Tobacco mosaic disease causes a distinctive mottling pattern on the leaves of tobacco plants, a mosaic of light and dark green patches. It spreads from plant to plant. It clearly behaves like an infection, and in the 1880s and 1890s, several scientists were trying to figure out what caused it. In 1892, Ivanovsky did something clever. He took sap from an infected tobacco plant and passed it through a Chamberland filter. Now, this filter is a key piece of the story, so let me explain what it actually is. Charles Chamberland, who worked in Pasteur's lab, had developed a porcelain filter with pores so tiny that bacteria couldn't pass through them. The filter was originally designed for purifying water, but it quickly became an essential tool in microbiology because it gave you a clean way to separate bacteria from the liquid they were swimming in. If you poured an infected fluid through a Chamberland filter, the bacteria would get trapped in the porcelain, and the liquid that came out the other side would be sterile. That was the whole point. So Ivanovsky filtered the sap from his diseased tobacco plants, expecting the filtered liquid to be harmless. But when he applied that filtered sap to healthy tobacco plants, they got sick anyway. The infectious agent, whatever it was, had passed right through a filter that should have stopped any known bacterium. Now, think about what that means for a moment. In 1892, the germ theory of disease was the most powerful new idea in medicine. Scientists had spent decades proving that bacteria caused disease, developing the tools to find them, the techniques to grow them, the logic to connect them to specific illnesses. And here was Ivanovsky, with an infectious agent that simply wasn't a bacterium. It was too small to see, too small to filter out. It couldn't be grown on any culture medium they had. By every tool available, it was invisible, and yet it was unquestionably real because it made plants sick. Ivanovsky himself didn't fully appreciate what he'd found. He suspected that maybe the filter had a defect, or that perhaps the infectious agent was a toxin produced by bacteria, some chemical that had passed through the filter rather than a living organism. He published his findings, but he didn't push the interpretation very far. It was a Dutch microbiologist named Martinus Beijerinck who, in 1898, repeated the filtration experiments and went further. Beijerinck showed that the infectious agent could be diluted and passed from plant to plant, and it would regain its full strength in each new host. That ruled out a simple toxin, because a toxin would get weaker with each dilution. Whatever this was, it was multiplying. It was reproducing inside the plant, but it couldn't reproduce on its own. It needed the living plant cells to do it. Beijerinck called it a contagium vivum fluidum, a contagious living fluid. He believed it was something fundamentally different from bacteria, something liquid and alive in a way that didn't fit any existing category. He was wrong about the fluid part. Viruses aren't liquid, they're particles. But he was profoundly right about the bigger insight. This was a new kind of entity, not a bacterium, not a toxin, something else entirely. And here's what's really fascinating about this moment. Beijerinck couldn't see the thing he was studying. He couldn't grow it in a dish. He couldn't kill it with the techniques that killed bacteria. He could only detect it by its effects, by what it did to the plants it infected. For the next several decades, that's essentially how all virus research worked. Scientists studied shadows on the wall. They knew something was there because of the damage it left behind, but the thing itself remained invisible. Around the same time, in 1898, Friedrich Loeffler and Paul Frosch in Germany showed that foot and mouth disease in cattle was also caused by a filterable agent, the first demonstration that these mysterious entities could infect animals, not just plants. And then, year by year, the list grew. Yellow fever, rabies, smallpox, polio, influenza, one by one, diseases that had plagued humanity for centuries, turned out to be caused by these tiny, invisible, filterable agents that could only reproduce inside living cells, but nobody could actually see one. Think about how strange that is. By the early 1900s, scientists knew that viruses existed. They knew viruses caused some of the most devastating diseases in human history. They were developing vaccines against some of them, and they had never once laid eyes on a single virus particle. The best microscopes of the era used visible light, and visible light has a fundamental physical limitation. The wavelength of visible light is roughly 400 to 700 nanometers, and you simply cannot resolve details smaller than about half the wavelength of the light you're using. Most viruses are somewhere between 20 and 300 nanometers across. They were physically below the resolution limit of any optical microscope that could ever be built. It's not that the microscopes weren't good enough. It's that the laws of physics made it impossible. So for decades, virologists worked in the dark, in a very literal sense. They developed incredibly ingenious, indirect methods. They measured how fast viruses settled in a centrifuge to estimate their size. They passed them through filters with different pore sizes to figure out roughly how big they were. They studied the diseases viruses caused, mapped how infections spread, developed vaccines and treatments, all without ever seeing the thing they were fighting. And then, in the 1930s, everything changed. A German physicist named Ernst Ruska, working with his colleague Max Knoll, developed the first electron microscope. Instead of using light, it used beams of electrons, which have wavelengths thousands of times shorter than visible light. Suddenly, you could see things at the scale of nanometers. And in 1935, an American biochemist named Wendell Stanley did something remarkable. He crystallized the tobacco mosaic virus. The same virus that Ivanovsky and Beijerinck had studied decades earlier. Stanley purified it, concentrated it, and it formed crystals like salt or sugar. This was shocking, because living things don't form crystals. Crystals are what minerals do, what chemicals do. The fact that a virus could be crystallized raised a question that, honestly, we're still wrestling with today. Are viruses alive? Stanley won the Nobel Prize in Chemistry for that work in 1946. And when electron microscopes finally captured images of the tobacco mosaic virus, scientists saw something elegant and strange. Long, thin rods, about 300 nanometers long and 18 nanometers wide, built from a spiral arrangement of identical protein subunits wrapped around a core of RNA. It was geometric. It was precise. It looked almost engineered, and it was so much simpler than any cell, so stripped down, so minimal, that it raised a deeper question than just what viruses are. It raised the question of what the minimum requirements for something to replicate itself actually look like. What's the least amount of biological machinery you need to make more of yourself? That question is going to stay with us tonight, because it turns out the answer is both simpler and stranger than anyone expected. To really understand it, we need to talk about what's actually inside a virus, what it's made of, how it's built, and why that architecture matters so much for how it does what it does. which is get inside your cells and hijack them. So let's start with the basics. A virus, at its simplest, is really just two things: a piece of genetic material and a protein shell that protects it. That's it. That's the minimum. You don't need a nucleus. You don't need mitochondria. You don't need ribosomes or a cell membrane or any of the thousands of molecular machines that even the simplest bacterium carries around. A virus is, in a sense, a message in a bottle. The message is the genetic instructions, the blueprint for making more viruses, and the bottle is the protein coat that keeps those instructions safe while the virus drifts through the world, waiting to find a cell it can infect. That protein coat has a name. It's called a capsid. And capsids are genuinely beautiful structures, not beautiful in a vague, poetic way, but beautiful in the way that a geodesic dome is beautiful, or a perfectly tiled floor. They're geometric. They follow mathematical rules, and the reason for that is elegant in itself. Viruses have very small genomes, right? They don't have a lot of genetic information to work with, so they can't encode instructions for hundreds of different structural proteins. Instead, most viruses encode just one or a few types of protein subunit, and then those subunits self-assemble into the capsid by repeating the same shape over and over again in a symmetrical pattern. It's like building a cathedral out of a single type of brick. You get complexity and strength from the arrangement, not from the diversity of the parts. There are really two main architectural plans that viruses use. The first is what you saw with the tobacco mosaic virus, helical symmetry. Picture a spiral staircase. The protein subunits stack in a helix, wrapping around the genetic material like the steps of that staircase, winding around a central pole. The result is a rod or a tube. It's simple. It's sturdy, and it can be as long as it needs to be. You just keep adding more subunits to the spiral. Tobacco mosaic virus is the classic example, but there are plenty of others. The Ebola virus, for instance, has that long thread-like shape that you've probably seen in micrographs. That's helical symmetry at work. Though Ebola's got some additional complexity we'll get to. The second major plan is icosahedral symmetry, and this one is everywhere. An icosahedron is a shape with 20 triangular faces, like a 20-sided die if you've ever played tabletop games. It's the most efficient way to enclose a volume using identical repeating subunits. And here's what's really clever about it. The math behind this was actually worked out in the early 1960s by Donald Caspar and Aaron Klug. Klug, by the way, went on to win the Nobel Prize in Chemistry in 1982, partly for his work on understanding virus structures. What Caspar and Klug showed was that you could build larger and larger icosahedral shells by using more copies of the same protein subunit, arranged according to specific geometric rules they called triangulation numbers. The basic icosahedron has 60 subunits, 3 per triangular face, 20 faces, 60 total. But if you want a bigger shell, you can use 180 subunits, or 240 or more, following these mathematical principles. It's like scaling up a dome while keeping the same architectural logic. Think about the viruses you've heard of. The common cold. Many of those are rhinoviruses, and they're tiny icosahedrons, maybe 30 nanometers across. Poliovirus, same deal. Adenoviruses, which cause some respiratory infections, are bigger icosahedrons with little antenna-like fibers sticking out from the vertices. Herpes viruses, much larger, but still fundamentally icosahedral at their core. This shape shows up again and again because it works, it's stable, it's economical, and it self-assembles, which is key. The protein subunits are designed so that when they bump into each other in the right conditions, they snap together like puzzle pieces. No external guidance needed. Now, some viruses don't fit neatly into either category. Bacteriophages, the viruses that infect bacteria, often look like tiny lunar landers. They have an icosahedral head that contains the DNA, a helical tail tube, and these spidery leg-like fibers at the base that they use to latch onto the bacterial surface. They're a combination of both symmetry types in one organism, if you want to call it an organism. When electron microscopy first revealed these structures in the 1940s and 50s, people were genuinely startled. They looked mechanical. They looked designed. There's a famous set of electron micrographs from the 1950s and 60s. Researchers like Thomas Anderson and later Eduard Kellenberger captured images of these phages that looked like something out of science fiction. But here's the thing. The capsid is just the outer shell. What matters most is what's inside, the genetic material. And this is where viruses get really interesting, because they're far more diverse in their genetic strategies than any other group of biological entities. Cells, all cells, whether bacterial, plant, animal, fungal, they all use double-stranded DNA as their primary genetic material. That's it. That's the universal standard. DNA gets transcribed into messenger RNA. Messenger RNA gets translated into protein. It's called the central dogma of molecular biology, and Francis Crick laid it out in 1958. Every cell on Earth follows it. Viruses don't care about your central dogma. Some viruses use double-stranded DNA, sure. Herpes viruses, pox viruses, adenoviruses, they're DNA viruses, and they play by roughly the same rules as cells. But plenty of viruses use single-stranded DNA instead. And then there are the RNA viruses, which is where things get wild. Some use double-stranded RNA. Some use single-stranded RNA that can be read directly by the cell's ribosomes, as if it were messenger RNA. These are called positive-sense RNA viruses, and they include things like poliovirus, hepatitis C, coronaviruses, and the common cold rhinoviruses. Others use single-stranded RNA that's the mirror image of messenger RNA, negative sense, and it has to be copied into the complementary strand before it can be read. Influenza, Ebola, measles, those are negative sense RNA viruses. And then there are the retroviruses, which do something that genuinely broke people's brains when it was discovered. HIV is the most famous example. A retrovirus carries its genome as single-stranded RNA, but once inside the cell, it uses a special enzyme called reverse transcriptase to convert that RNA back into DNA. DNA made from an RNA template. That's the reverse of the central dogma, which is why they're called retroviruses. And then, and this is the part that still gives me chills, that DNA copy gets inserted directly into the host cell's own genome. It becomes part of your chromosomes. Your cells will copy it every time they divide, and they may carry it forever. In fact, something like 8% of the human genome is made up of ancient retroviral sequences, remnants of infections that happened millions of years ago in our ancestors that got passed down through the generations because they ended up in the DNA of egg or sperm cells. 8%. That's more of your genome than the part that codes for proteins. You are, in a very real molecular sense, part virus. This incredible diversity in genetic strategy was eventually organized into a classification system by a David Baltimore, a virologist who won the Nobel Prize in 1975 for his co-discovery of reverse transcriptase. The Baltimore classification divides all viruses into 7 groups based on how they produce messenger RNA from their genomes. It's not based on what shape they are, or what they infect, or what diseases they cause. It's based purely on their replication strategy, and it's still the most useful framework we have for understanding how viruses work at the molecular level. Now, I mentioned that some viruses have an additional layer beyond the capsid, and this is worth spending a moment on because it matters a lot for how certain viruses behave. Some viruses, influenza, HIV, coronaviruses, Ebola, herpes viruses, are wrapped in a lipid envelope. This is essentially a piece of cell membrane that the virus steals from its host cell as it buds out. It's a fatty, flexible outer coat studded with viral proteins that stick out like spikes or mushrooms. Those surface proteins are critically important because they're what the virus uses to recognize and attach to host cells. The spike protein of SARS-CoV-2, the coronavirus that causes COVID-19, is an envelope protein. It's what grabs onto a receptor called ACE2 on the surface of your cells. The hemagglutinin protein on the surface of influenza viruses is what gives flu strains their H number, H1N1, H5N1, and so on. Here's a practical detail that connects directly to your everyday life. That lipid envelope is fragile. It's made of fat, basically, and soap destroys fat. That's why washing your hands with soap is so effective against enveloped viruses like influenza and coronaviruses. The soap dissolves the envelope. The virus falls apart, and it's no longer infectious. Non-enveloped viruses like norovirus, the stomach bug, are tougher. They don't have that fatty outer layer to dissolve, so they can survive on surfaces longer, and they're harder to kill with simple soap. That's why norovirus outbreaks on cruise ships are so notoriously difficult to control. The virus is basically a naked protein shell, and it's remarkably resilient. So you've got this picture now. A virus is a package, genetic material on the inside, a protein capsid around it, sometimes a lipid envelope around that, with surface proteins that determine what the virus can attach to. It's minimal, it's elegant, and it's completely inert on its own. A virus sitting on a doorknob or floating in a droplet of moisture isn't doing anything. It's not metabolizing, it's not growing, it's not responding to its environment in any active way. It's just a particle, a very sophisticated particle, but a particle nonetheless. Everything changes the moment it encounters the right cell, and the process by which a virus gets into a cell takes over its machinery and turns it into a virus factory. That process is one of the most intricate and, honestly, one of the most unsettling things in all of biology. It starts with recognition, with that first molecular handshake between a protein on the virus surface and a receptor on the cell, and the specificity of that interaction, the precision of that lock and key fit, is what determines everything about which species a virus can infect, which tissues it targets, and ultimately what kind of disease it causes. So let's stay right there at that moment of contact, because what happens next is genuinely remarkable. Think about what the virus needs to accomplish. It's on the outside of a cell, and it needs to get its genetic material, its RNA or DNA, to the inside, past a membrane that exists specifically to keep foreign things out. The cell membrane is a lipid bilayer, a double layer of fat molecules with their water-loving heads facing outward and their water-fearing tails tucked inside. And it's actually a pretty formidable barrier. It's not a wall exactly. It's more like a fluid, shifting surface with proteins embedded in it like icebergs in a sea. And the cell is very selective about what it lets through. Small molecules, sure. Ions through specific channels, yes. But a whole virus? That's not supposed to happen. And yet viruses have evolved extraordinarily clever ways to get past that barrier. And the method depends entirely on the type of virus we're talking about. Let's start with enveloped viruses, because their approach is, in a way, the most elegant. Remember, an enveloped virus has its own lipid membrane, stolen from a previous host cell, surrounding its capsid. So you've got two membranes facing each other, the virus's envelope and the cell's plasma membrane. And what some enveloped viruses do is fuse those two membranes together. They literally merge. The viral envelope and the cell membrane become one continuous sheet of lipid, and in the process, the contents of the virus, the capsid, the genome, get dumped into the interior of the cell. It's like two soap bubbles touching and becoming one. The influenza virus does a version of this, but with a twist that's worth slowing down for, because it reveals just how precisely engineered these molecular machines are. When influenza binds to a cell, using its hemagglutinin protein latching onto sialic acid residues on the cell surface, the cell doesn't just let the virus in through the front door. Instead, the cell actually engulfs the virus. It pulls it inward in a little pocket of membrane, creating a small bubble called an endosome. So now the virus is inside the cell, but it's still trapped inside this membrane-bound compartment. It hasn't actually reached the cytoplasm yet. Here's where things get clever. As the endosome matures, the cell pumps hydrogen ions into it, making the interior more acidic. The pH drops, and that drop in pH triggers a dramatic conformational change in the hemagglutinin protein. The protein literally snaps into a new shape, exposing a previously hidden segment called the fusion peptide, which is essentially a molecular harpoon. It shoots into the endosomal membrane and pulls the viral envelope and the endosomal membrane together until they fuse. The capsid spills out into the cytoplasm. The virus has escaped its trap by using the cell's own acidification process as a trigger. That's not accidental. That's the product of millions of years of evolutionary refinement, a molecular mechanism so precisely tuned to a specific pH that you can actually measure the exact acidity at which it fires, around pH 5 to 5.5 for most influenza strains. And this, by the way, is exactly what the drug amantadine was designed to interfere with. Amantadine blocks an ion channel called M2 on the influenza A virus that helps acidify the interior of the virus particle itself, which is part of the uncoating process. Block that channel, and the virus can't properly release its genome even after fusion occurs. It was one of the first antiviral drugs, approved back in 1966, and it worked until resistance mutations in the M2 channel became so widespread that by 2006 or so, the CDC stopped recommending it for most influenza strains. The virus simply evolved a slightly different version of the M2 protein that amantadine couldn't bind to anymore. And that right there tells you something important about the challenge of antiviral medicine, which we'll come back to later. But for now, the point is that understanding the precise molecular mechanism of viral entry isn't just academic. It's the foundation for designing drugs that can stop the process. Non-enveloped viruses have a different problem. They don't have a lipid envelope to fuse with the cell membrane, so they have to find another way through. Some of them, like poliovirus, bind to their receptor. In polio's case, a protein called CD155, and the binding event itself triggers a change in the capsid structure that opens up a pore in the cell membrane. The viral RNA gets threaded through that pore directly into the cytoplasm, while the empty capsid stays outside, stuck to the cell surface like a discarded syringe. Others get pulled into endosomes, similar to influenza, but then they use the acidic environment to partially disassemble their capsid, punching holes in the endosomal membrane and escaping into the cytoplasm. Adenoviruses do something particularly dramatic. They actually lyse the endosome, bursting it open from the inside using a protein called protein 6 that has membrane-disrupting properties. The virus essentially blows its way out of the compartment. So the virus has gotten in. Its genetic material is now loose inside the cell. And this is where the real takeover begins. And honestly, this is the part that I find most fascinating, because it reveals just how deeply viruses have learned to exploit the fundamental machinery of life. Every cell in your body is essentially a protein-making factory. That's what cells do. They read the instructions encoded in DNA, transcribe those instructions into messenger RNA, and then ribosomes translate that messenger RNA into proteins, DNA to RNA to protein. That's the central dogma of molecular biology, as Francis Crick articulated it in 1958. And what a virus does at its core is hijack that production line. It slips its own instructions into the queue and tricks the cell's ribosomes into making viral proteins instead of, or in addition to, the cell's own proteins. But how this happens depends entirely on what kind of genetic material the virus brought with it. And this is where David Baltimore's classification system really pays off, because each of those 7 groups has a fundamentally different replication strategy. Let's start with the simplest case conceptually. A positive sense, single-stranded RNA virus, Baltimore Group 4, carries a genome that's essentially already a messenger RNA. The moment that RNA hits the cytoplasm, the cell's ribosomes can grab onto it and start translating it into proteins immediately. No intermediate steps required. It's like handing someone a document that's already in their language. They can read it right away. Poliovirus works this way. So does SARS-CoV-2. So does hepatitis C. The viral RNA walks in the door and immediately starts issuing orders. Among the first proteins it produces are the enzymes the virus needs to copy its own RNA, an RNA-dependent RNA polymerase, an enzyme that cells don't normally have because cells don't normally need to copy RNA from an RNA template. This enzyme is error-prone, by the way. It doesn't have the proofreading mechanisms that DNA polymerases have, which means RNA viruses tend to mutate fast, really fast, and that's a big deal, which we'll get into. Now, negative sense single-stranded RNA viruses, Group V, have the opposite problem. Their genome is the complement of messenger RNA. It's like a mirror image, or like having a document written in a code that needs to be decoded before anyone can read it. The cell's ribosomes can't do anything with it directly, so these viruses have to bring their own RNA-dependent RNA polymerase along with them, packaged inside the virus particle, ready to go. The moment the genome enters the cell, this enzyme starts converting the negative sense RNA into positive sense messenger RNA that the ribosomes can then translate. Influenza does this, Ebola does this, rabies does this, and here's a subtle but important consequence. Because these viruses need to bring their own polymerase, they tend to be slightly larger and more complex than positive sense RNA viruses. They're carrying more equipment. Retroviruses, group 6, do something that genuinely shocked the biological world when it was discovered. Howard Temin at the University of Wisconsin had been arguing since the early 1960s that certain RNA tumor viruses must be copying their RNA into DNA. That information was flowing backward from RNA to DNA, violating what everyone thought was an inviolable rule. People thought he was wrong. Some thought he was a little crazy. The central dogma said information flows from DNA to RNA to protein, period. But Temin was right. In 1970, both he and David Baltimore, working independently at different institutions, simultaneously discovered reverse transcriptase, the enzyme that copies RNA into DNA. It was one of those moments in science where the world shifts. Temin and Baltimore shared the Nobel Prize in 1975 for it, along with Renato Dulbecco. What HIV does, what all retroviruses do, is convert their RNA genome into double-stranded DNA using reverse transcriptase, and then that DNA gets inserted into the host cell's own chromosomes by another viral enzyme called integrase. Think about what that means. The virus's genetic instructions become part of your DNA. They're woven into your genome, and every time that cell divides, it copies the viral DNA right along with its own. The virus has essentially become a permanent part of the cell's identity. This is why HIV is so difficult to cure. You can suppress the virus with antiretroviral drugs. You can drive it down to undetectable levels in the blood, but those integrated copies are still sitting there in long-lived memory T cells, silent, waiting. If you stop treatment, the virus can reactivate from those reservoirs. It's hiding in the one place you can't easily reach, inside the cell's own instruction manual. And remember what we talked about earlier, about roughly 8% of the human genome being made up of ancient retroviral DNA. Those are the remnants of infections that happened millions of years ago in our evolutionary ancestors. Retroviruses that infected germline cells, eggs or sperm, and got passed down through generations. Most of those sequences are broken now, mutated beyond function, but some of them have actually been co-opted by our own biology. There's a protein called syncytin that's essential for the formation of the placenta in mammals, and it's derived from an ancient retroviral envelope gene. A virus gave us a protein that makes pregnancy possible. That's one of those facts that just stops you in your tracks when you really think about it. But let's come back to the replication process, because once the viral proteins start getting made, the cell transforms into something it was never meant to be. The virus needs copies of its genome, and it needs capsid proteins. And if it's an enveloped virus, it needs to arrange for its envelope too. The cell's own protein synthesis gets suppressed or outcompeted. Resources get diverted. In some cases, the virus actively shuts down the host cell's ability to make its own proteins. Poliovirus, for instance, cleaves a protein called EIF4G that's essential for the cell's normal translation machinery, effectively silencing the cell's own messages, while the viral RNA, which uses a different mechanism to recruit ribosomes, keeps getting translated. It's ruthless, it's targeted, and it's effective. As viral proteins accumulate and new copies of the genome are made, the components start assembling into new virus particles. This is called assembly, and in many cases, it's a remarkably self-directed process. The capsid proteins have built-in instructions in their very shape and charge distribution to spontaneously come together into the correct structure. Tobacco mosaic virus was actually one of the first systems where this was demonstrated. Heinz Frankel-Conrat and Robley Williams showed in 1955 that if you separated the RNA and the coat protein of TMV and then mixed them back together in a test tube, they would spontaneously reassemble into functional infectious virus particles. No cellular machinery needed, no enzymes, just the intrinsic properties of the molecules themselves, self-assembly. It was a stunning demonstration that the information for building the virus was encoded in the components themselves. And then comes the final act, release. The new virus particles need to get out of the cell and go find new cells to infect. And this happens in different ways depending on the virus. Some viruses are brutal about it. They replicate until the cell is so packed with new particles that it simply bursts open, lyses, spilling hundreds or thousands of new viruses into the surrounding tissue. This is what happens with many non-enveloped viruses. It's efficient but destructive, and it's a big part of why viral infections cause tissue damage and inflammation. Enveloped viruses often take a more subtle approach. They bud. The viral capsid, assembled inside the cell, migrates to the cell membrane, where viral envelope proteins have already been inserted. Capsid pushes against the membrane from the inside, and the membrane wraps around it, pinching off to form a new enveloped virus particle. The virus literally steals a piece of the cell's own membrane as it leaves. The cell doesn't necessarily die right away. It might keep producing virus particles for a while, a slow leak rather than an explosion. HIV does this. Influenza does this. And this budding process is where the neuraminidase protein on influenza becomes critical, because newly Budded virus particles tend to stick to the cell surface. Their hemagglutinin binds to the same sialic acid residues that got them in. Neuraminidase clips those sialic acid molecules, freeing the new viruses to drift away and infect other cells. And that's exactly what the drugs oseltamivir and zanamivir, Tamiflu and Relenza target. They inhibit neuraminidase, so new virus particles stay stuck to the cell surface and can't spread efficiently. It doesn't kill the virus directly. It just slows the release, giving your immune system time to catch up, which brings us to the immune system. Because this whole time we've been talking about the virus's side of the story, how it gets in, how it replicates, how it gets out. But your body isn't just sitting there passively while all this happens. The moment a virus enters your body, it triggers a cascade of defensive responses that are every bit as complex and fascinating as the virus itself. And the interplay between viral attack and immune defense, that evolutionary arms race that's been going on for hundreds of millions of years, is really where the deepest stories in virology live, because viruses haven't just evolved to infect cells. They've evolved to evade, deceive, and sometimes directly sabotage the immune system. And the immune system, in turn, has evolved countermeasures to those evasions. It's layer upon layer of strategy and counter-strategy. And the first line of defense, the one that kicks in within minutes of infection, is something called the innate immune system. And it's ancient. We're talking about defenses that evolved long before anything resembling an antibody or a T cell existed. Insects have versions of this system. Plants have their own parallel version. It's the deep, old machinery of self-defense, and it's remarkably good at what it does. So let's say a virus. Let's use influenza again, since we've been following it. Let's say it gets into your respiratory tract, latches onto an epithelial cell lining your airway, fuses its way in, and starts replicating within that very cell before any immune cell even knows something is wrong. There's already a detection system at work, and it starts with molecules called pattern recognition receptors. Think of them as tripwires. They're proteins sitting inside your cells and on their surfaces, and they're tuned to detect molecular signatures that don't belong, things that are common to pathogens, but that your own healthy cells don't normally produce. These signatures are called pathogen-associated molecular patterns, or PAMPs, and for viruses, one of the biggest red flags is double-stranded RNA. Here's why that matters. Remember how we talked about positive-sense single-stranded RNA viruses needing to make a complementary strand to replicate? During that process, you get transient stretches of double-stranded RNA, a positive strand paired with a negative strand, and your cells almost never produce long double-stranded RNA on their own. So when a receptor inside your cell encounters double-stranded RNA, it's essentially catching the virus in the act of replication. One of the key receptors that does this is called RIG-I, retinoic acid inducible gene 1. It was identified in the early 2000s, and it patrols the cytoplasm looking for exactly this kind of foreign RNA. When RIG-I grabs onto double-stranded RNA, it triggers a signaling cascade that converges on something genuinely important: the production of interferons. Interferons. The name tells you what they do. They interfere. They were discovered back in 1957 by Alec Isaacs and Sheen Lindenman, two researchers working at the National Institute for Medical Research in London. And the experiment was beautifully simple in concept. They exposed chicken cells to inactivated influenza virus that couldn't replicate, and found that those cells released a substance into the surrounding fluid, took that fluid, and applied it to fresh cells. Those fresh cells became resistant to live influenza infection. Something the first cells had secreted was protecting the second cells. They called it interferon, and it took decades to fully work out what was happening. But the basic insight was right. Infected cells send out a molecular alarm signal, and neighboring cells that receive that signal go into a kind of defensive lockdown. Here's how it works in a bit more detail. When RIG-I or another sensor like MDA5 detects viral RNA, it activates transcription factors, particularly one called IRF3 and another you've probably heard of, NF-kappa B. These switch on the genes for type 1 interferons, mainly interferon alpha and interferon beta. The cell secretes these interferons, and they drift outward and bind to receptors on neighboring cells, cells that aren't infected yet. When interferon lands on those receptors, it triggers the JAK-STAT signaling pathway inside the receiving cell, which turns on hundreds of genes collectively called interferon-stimulated genes, or ISGs. And what these genes do is essentially prepare the cell for war. They produce proteins that degrade viral RNA, proteins that shut down protein synthesis so the virus can't use the cell's ribosomes, proteins that prevent viral entry, proteins that block viral assembly. The cell becomes a fortress. It's not immune. A determined virus can still get in, but the odds shift dramatically. And think about what this means from the virus's perspective. You've just spent all this effort getting into one cell, hijacking its machinery, making copies of yourself, budding out into the extracellular space, and now the cells all around your target are armored up and waiting for you. That's a serious problem, which is exactly why so many viruses have evolved specific countermeasures against the interferon system. This is where the arms race gets really interesting. Influenza, for instance, has a protein called NS1, non-structural protein 1. And one of its main jobs is to block the interferon response. NS1 can bind to double-stranded RNA and hide it from RIG-I. It can also directly inhibit the processing and export of host cell messenger RNA. which means the cell struggles to actually produce and secrete interferon, even if the alarm has been triggered. Different influenza strains have NS1 proteins of varying potency, and this is one of the reasons some flu strains are more virulent than others. The 1918 pandemic influenza, the strain that killed somewhere between 50 and 100 million people, when researchers reconstructed that virus in 2005 in a high-security lab at the CDC, one of the things they found was that its NS1 was exceptionally good at suppressing the interferon response. The virus could replicate almost unchecked in the early hours of infection, building up to enormous numbers before the immune system could mount an effective defense. And influenza is far from the worst offender. Ebola virus carries a protein called VP35 that blocks RIG-I signaling, and another called VP24 that specifically prevents STAT1 from entering the nucleus. Remember, STAT1 is part of that JHEC-STAT pathway that interferon activates. So even if neighboring cells receive the interferon signal, VP24 prevents them from actually responding to it. The alarm goes off, but the fortress doors don't close. That's part of why Ebola can be so devastating. It essentially blinds the innate immune system during the critical early window, when containment might still be possible. Now, interferons aren't the only thing the innate immune system throws at viruses. Let's talk about the cells that come rushing in, the first responders, so to speak. Within hours of infection, you get an influx of innate immune cells to the site, and the most important early arrivals are natural killer cells, or NK cells. These are lymphocytes, but they're not part of the adaptive immune system. They don't need to be trained against a specific virus. They're born ready. Here's the clever thing about how NK cells decide what to kill. Your healthy cells constantly display molecules on their surface called MHC class 1, Major Histocompatibility Complex class 1. These molecules are like identity badges. They present little fragments of whatever proteins the cell is making, and NK cells check those badges. If a cell is displaying normal MHC class 1, the NK cell gets an inhibitory signal. This cell is fine, move along. But many viruses, as part of their strategy to hide from the adaptive immune system, downregulate MHC class 1 on the surface of infected cells. They're trying to become invisible to T cells, which we'll get to. But in doing so, they accidentally make themselves visible to NK cells. The NK cell comes along, checks for the badge, doesn't find it, and interprets the absence as a sign of infection. It then kills the cell by releasing perforin and granzymes. Perforin punches holes in the cell membrane, and granzymes enter through those holes and trigger apoptosis, programmed cell death. The cell dismantles itself in an orderly way, taking the virus factory down with it. It's a beautiful system. The virus tries to hide from one branch of immunity and inadvertently exposes itself to another. But of course, Some viruses have found ways around even this. Cytomegalovirus, CMV, a herpes virus that infects a huge percentage of the global population, produces decoy MHC-like molecules that sit on the cell surface and fool NK cells into thinking everything is normal. It's mimicry at the molecular level. The virus has essentially stolen a piece of the immune system's own language and is using it to lie. There's another innate defense worth mentioning here, and it's one that's been in the news a lot in recent years: the inflammatory response. When cells detect a virus, they don't just produce interferons. They also release cytokines and chemokines, signaling molecules that recruit more immune cells to the area and ramp up inflammation. Inflammation gets a bad reputation, and honestly, when it goes wrong, it deserves that reputation. But in its normal form, inflammation is essential. It increases blood flow to the infected area, makes blood vessel walls more permeable so immune cells can squeeze through into the tissue, and creates an environment that's hostile to the pathogen. The problem, and this is a big problem, is that sometimes the inflammatory response overshoots. Instead of a controlled, localized fire, you get a conflagration. This is what's sometimes called a cytokine storm, and it's been implicated in the worst outcomes of several viral infections. In the 1918 flu pandemic, many of the deaths weren't caused directly by the virus destroying lung tissue. They were caused by the immune system's own inflammatory response spiraling out of control, flooding the lungs with fluid and immune cells, essentially drowning the patient from within. The same phenomenon was seen in severe cases of SARS in 2003, in H5N1 avian influenza, and in COVID-19. Young, healthy adults with strong immune systems were sometimes hit hardest, precisely because their robust immune response produced a more violent inflammatory reaction. It's a terrible irony. The very strength of the defense becomes the weapon that does the most damage, and this is something that researchers have grappled with for years. How do you help the immune system fight the virus without letting it destroy the host? Dexamethasone, a corticosteroid that suppresses inflammation, became one of the most important treatments for severe COVID-19, not because it did anything to the virus, but because it dialed down the immune system's self-destructive overreaction. It was the recovery trial in the UK, results announced in June 2020, that demonstrated this clearly. Dexamethasone reduced mortality in patients who needed oxygen or ventilation, a cheap, decades-old anti-inflammatory drug, saving lives not by attacking the virus, but by restraining the body's own defenses. So the innate immune system is this fast, broad, ancient layer of defense. It buys time. It slows the virus down, sounds the alarm, recruits reinforcements, and in many cases, it's enough on its own to clear a minor infection before you ever notice symptoms. But for serious viral infections, the ones that make you sick, the ones that can kill. The innate system needs backup. It needs the adaptive immune system, the precision-guided, highly specific, memory-forming branch of immunity that takes days to spin up, but, once it does, can target a virus with extraordinary accuracy. And the way the innate system hands off to the adaptive system, the bridge between those two worlds, involves some of the most elegant cell biology you'll ever encounter. It centers on a type of cell called the dendritic cell. And what dendritic cells do is essentially this. They act as scouts and messengers. Dendritic cells are stationed throughout your body, in your skin, in the lining of your lungs, in your gut, basically anywhere a virus is likely to first make contact. And they're constantly sampling their environment, reaching out with these long branching extensions. That's actually where the name comes from, dendrite, from the Greek word for tree, because under a microscope they look like little trees with all these spindly arms stretching outward. They're tasting the world around them, pulling in bits and pieces of whatever's floating by, breaking those pieces down, and checking whether any of it looks dangerous. Now, when a dendritic cell encounters a virus, or, more often, the debris of a virus that's already been partially dealt with by the innate response, something remarkable happens. The dendritic cell doesn't just destroy the virus and move on. It takes fragments of the viral proteins, these short sequences of amino acids called peptides, and it loads them onto special molecules on its surface. These molecules are called MHC class II molecules, and they work almost like little display shelves. The dendritic cell is essentially saying, "Look what I found." It takes these viral fragments and presents them on its surface for other immune cells to inspect. And here's where the journey gets beautiful. Once a dendritic cell has captured viral material and loaded it onto MHC class II, it changes behavior completely. It stops being a sentinel sitting in the tissue and becomes a traveler. It leaves the site of infection, enters the lymphatic system, that network of vessels and nodes that runs through your whole body, and it migrates to the nearest lymph node. Think of lymph nodes as these little meeting halls scattered throughout your body, places where immune cells congregate and exchange information. You've probably felt them swell up in your neck or under your jaw when you're fighting off a cold. That swelling, that's the immune system holding a war council. The dendritic cell arrives at the lymph node, carrying its cargo of viral peptides displayed on MHC class II, and it starts doing something that immunologists call antigen presentation. It's showing those viral fragments to T cells, specifically to a type of T cell called a helper T cell, or CD4 positive T cell. Now, here's the thing that makes this system so extraordinary. You have millions and millions of T cells in your body, and each one has a slightly different receptor on its surface. Each T cell receptor is shaped to recognize one specific peptide MHC combination, just one. So the dendritic cell is essentially walking through a crowd of millions of unique T cells, holding up its little display of viral fragments, waiting for the one T cell, maybe 1 in a million, maybe 1 in 10 million, whose receptor happens to fit that particular viral peptide like a key in a lock. Wound amound swamped, and when that match happens, when a helper T cell's receptor clicks into place against the peptide being presented by the dendritic cell, that T cell wakes up, it activates, it starts dividing rapidly, cloning itself over and over, producing an army of identical helper T cells that all recognize that same viral fragment. This process is called clonal expansion, and it's one of the most important concepts in immunology. Your body doesn't keep huge, standing armies of T cells against every possible threat. That would be impossibly expensive in biological terms. Instead, it keeps tiny populations of T cells with an enormous diversity of receptors, and when one of them finds its match, it multiplies explosively. You go from maybe a handful of cells that can recognize a particular virus to millions of them within days. Thiever denaf iller. Those activated helper T cells then do exactly what their name suggests. They help. They release signaling molecules, cytokines again, that coordinate and amplify the rest of the adaptive immune response. They help activate another crucial type of cell, B cells. And B cells are the ones that make antibodies. Let's talk about antibodies for a moment, because they're one of those things everyone's heard of, but the actual mechanism is worth understanding. An antibody is a Y-shaped protein. Each one is designed to bind to a very specific molecular shape called an epitope on the surface of a pathogen. When a B cell gets activated, with help from those helper T cells we just talked about, it starts churning out antibodies at an astonishing rate. A single activated B cell, once it matures into what's called a plasma cell, can secrete thousands of antibody molecules per second. These antibodies flood into the blood and tissues, and they do several things. They can coat a virus particle directly, binding to the proteins on its surface in a way that physically blocks the virus from attaching to host cells. This is called neutralization, and it's exactly what it sounds like. The virus is neutralized, rendered harmless, unable to get in. Antibodies can also tag virus particles for destruction by other immune cells, a process called opsonization, which comes from a Greek word meaning to prepare for dining. Essentially, the antibody marks the virus as a meal for macrophages and other phagocytes. And here's where the concept of antigenic specificity becomes really practical. Think about why you get a flu shot every year, but you only needed 1 or 2 doses of the measles vaccine as a child. Measles is antigenically stable. The surface proteins that antibodies target don't change much from year to year. So once your immune system has seen measles, either through infection or vaccination, those antibodies and memory cells remain effective for decades. Influenza, on the other hand, is constantly changing. Its surface proteins, hemagglutinin and neuraminidase, those same molecules we talked about earlier, undergo two kinds of change. Antigenic drift is the slow, continuous accumulation of small mutations in those surface proteins. Every flu season, the virus is slightly different from last year's version, just different enough that last year's antibodies don't fit quite as well. It's like someone subtly reshaping the lock so your old key doesn't turn as smoothly. Then there's antigenic shift, which is much more dramatic. This happens when two different influenza strains infect the same cell and swap entire gene segments. Suddenly, you get a virus with a completely new hemagglutinin subtype, something the human population has never encountered before. That's how pandemic strains emerge. The 1918 H1N1, the 1957 H2N2, the 1968 H3N2, the 2009 H1N1. Each of those pandemic viruses carried a hemagglutinin that was fundamentally new to most people's immune systems. But let's come back to the other arm of the adaptive response, because antibodies are only half the story. The other half is cellular immunity, and it's carried out by a different type of T cell, the cytotoxic T cell, also called a CD8 positive T cell, or sometimes just a killer T cell. If antibodies are about finding and neutralizing free-floating virus particles in the blood and tissue fluids, killer T cells are about finding and destroying cells that have already been infected, and the mechanism here is elegant in a slightly terrifying way. Remember MHC class I molecules? We touched on those when we talked about NK cells and how they check for that self signal. Well, MHC class I does something else that's absolutely critical. Every nucleated cell in your body is constantly taking samples of the proteins being made inside it, chopping them into small peptides, and displaying those peptides on MHC class I molecules on the cell surface. It's like a window into what's happening inside the cell. Under normal conditions, the peptides on display are just fragments of your own normal proteins, and the immune system ignores them. But when a cell is infected with a virus, it's also producing viral proteins, because the virus has hijacked the cell's machinery to make copies of itself, and some of those viral protein fragments end up on MHC class I, displayed on the cell surface for all to see. A killer T cell patrols the body, checking MHC class I molecules on every cell it encounters. When it finds a cell displaying a viral peptide that matches its receptor, it knows that cell is infected, and it kills it. It releases perforin and granzymes, the same molecular weapons NK cells use, punching holes in the infected cell's membrane and triggering apoptosis, programmed cell death. The infected cell dies in an orderly way, taking the virus factory inside it offline before those new virus particles can be assembled and released. Think about what that means for a moment. Your body is willing to sacrifice its own cells to stop a viral infection. It's a calculated trade-off. Lose some cells now, or lose the whole organism later. And the precision of this system is remarkable. The killer T cell targets only cells displaying that specific viral peptide, leaving neighboring healthy cells untouched. It's surgical. Now, there's one more piece of this puzzle that might be the most important of all, and it's the reason you don't get chickenpox twice. After the infection is cleared, after the antibodies have neutralized the free virus, after the killer T cells have destroyed the infected cells, after the whole massive immune response winds down, most of those billions of cloned immune cells die off. They're not needed anymore, and maintaining them would be metabolically costly. But not all of them die. A small fraction of both T cells and B cells convert into memory cells. These are long-lived cells that persist in your body for years, sometimes for the rest of your life, carrying the molecular memory of that specific virus. And if that same virus ever shows up again, the response is dramatically faster. Instead of taking a week or more to find the right T cell, activate it, clone it, and mount a full response, your memory cells can kick into action within hours. The virus barely gets a foothold before the immune system crushes it. This is why second infections are often milder or even completely asymptomatic. Your immune system remembers, and it responds with overwhelming speed and force. This is also, of course, the entire principle behind vaccination. Edward Jenner didn't know any of this immunology when he used cowpox to protect against smallpox in 1796. He had no idea about T cells or B cells or MHC molecules or clonal expansion. He just observed that milkmaids who'd had cowpox didn't get smallpox, and he had the insight and the audacity to test whether deliberately exposing someone to the milder disease could protect them from the deadly one. But what he was actually doing, in modern terms, was presenting the immune system with antigens similar enough to smallpox that memory cells would be generated. Memory cells that would recognize and respond to the real virus if it ever appeared, and that insight that you could train the immune system in advance, that you could give it a preview of a threat it hadn't encountered yet. That idea has saved more human lives than almost any other single concept in the history of medicine. The numbers are staggering. Smallpox alone killed an estimated 300 million people in the 20th century before it was eradicated through vaccination in 1980. Polio, measles, diphtheria, tetanus, pertussis. The list of diseases brought under control through vaccination is extraordinary. But the story of how vaccines evolved from Jenner's crude cowpox inoculations to the sophisticated mRNA platforms used against COVID-19 is itself a fascinating journey through virology, and it involves some of the most creative problem solving in all of medicine and engineering, because that's really what vaccine development is. It's engineering. You're engineering an encounter between the immune system and something that looks enough like a pathogen to trigger all those beautiful cascading responses we just talked about. The dendritic cells grabbing antigens, the helper T cells coordinating, the B cells churning out antibodies, the memory cells settling in for the long haul, but without actually causing the disease. That's the trick. That's always been the trick, and figuring out how to pull it off safely and reliably for different viruses has taken more than 2 centuries of trial, error, heartbreak, and brilliance. So let's walk through it. After Jenner, the next giant leap came from Louis Pasteur. And yes, he keeps showing up in this story because the man was everywhere in 19th century microbiology. In the 1870s and 1880s, Pasteur was working with chicken cholera, a bacterial disease, and he stumbled onto something remarkable, almost by accident. He'd left a culture of the bacteria sitting out over a summer holiday when he came back and injected chickens with the old, weakened culture. They got mildly sick, but recovered. And then, here's the key part. When he later exposed those same chickens to a fresh, virulent culture, they didn't get sick at all. The weakened bacteria had trained their immune systems. Pasteur recognized what had happened immediately, and he was generous enough to name the technique vaccination in honor of Jenner's earlier work with vaccinia, the cowpox virus. What Pasteur had discovered was attenuation, the idea that you could weaken a pathogen, reduce its ability to cause disease, while still preserving its ability to provoke an immune response. And he went on to develop attenuated vaccines for anthrax and, most famously, rabies. His rabies vaccine, first used on a human in 1885, a 9-year-old boy named Joseph Meister, who'd been badly bitten by a rabid dog, was a sensation. The boy survived. Pasteur became one of the most celebrated scientists in the world. But attenuation was an art more than a science in those early days. You were essentially trying to cripple a pathogen just enough to make it harmless, but not so much that the immune system wouldn't bother responding to it. And the methods were crude, passing the pathogen through different animal hosts, growing it in unusual conditions, hoping that mutations would accumulate that weakened it. There was no way to precisely control what you were doing at the genetic level. You were rolling the dice and checking the results. Sometimes it worked beautifully, sometimes it didn't. The 20th century brought 2 major approaches to vaccine design, and both of them shaped modern medicine profoundly. The first was the live attenuated vaccine, a direct descendant of Pasteur's approach, but refined dramatically. The second was the inactivated vaccine, where you kill the pathogen entirely and inject the corpse, hoping the immune system will still recognize the surface proteins and generate a response. Each approach has strengths and trade-offs, and the history of how they were developed for specific viruses tells you a lot about the challenges of virology. Take polio. We talked earlier about how poliovirus enters cells, how it hijacks the ribosome, how it cleaves EIF4G to shut down host protein synthesis. It's a nasty, efficient little virus. And in the first half of the 20th century, it was terrifying. Summer after summer, outbreaks would sweep through communities, paralyzing children, filling hospital wards with iron lungs. Franklin Roosevelt contracted it in 1921, and his struggle with the disease helped galvanize public support for research. The March of Dimes, originally called the National Foundation for Infantile Paralysis, raised enormous sums from ordinary Americans, literally collecting dimes to fund polio research. Two scientists emerged as the central figures in the race for a polio vaccine, and they took opposite approaches. Jonas Salk, working at the University of Pittsburgh, developed an inactivated vaccine. He grew poliovirus in monkey kidney cells, then killed it with formaldehyde. The dead virus couldn't replicate, couldn't cause disease, but its surface proteins were intact enough to provoke an immune response. Salk's vaccine was tested in one of the largest clinical trials in history, the 1954 Francis Field trial, named after Thomas Francis Jr., who ran it, involving nearly 1.8 million children. When the results were announced on April 12, 1955, church bells rang. People wept in the streets. Salk became a national hero overnight. But Albert Sabin, a Polish-American researcher, believed a live attenuated vaccine would be superior. His reasoning was sound. A live virus, even a weakened one, would replicate in the gut, mimicking a natural infection more closely. It would provoke not just antibody production in the blood, but also mucosal immunity in the intestinal lining, right where poliovirus actually enters the body. And because the attenuated virus could spread from vaccinated individuals to their contacts, it could provide indirect protection to unvaccinated people in the community. Sabin's oral polio vaccine, given as drops on a sugar cube, which was brilliant from a logistics standpoint, no needles required, was licensed in the early 1960s and became the dominant polio vaccine worldwide for decades. Here's the thing, though. The Sabin vaccine had a problem, and it's a problem that illuminates one of the fundamental tensions in vaccine engineering. Because the attenuated virus was alive and replicating, it could, in very rare cases, mutate back toward virulence. About 1 in every 2.4 million doses could cause vaccine-associated paralytic poliomyelitis. 1 in 2.4 million sounds tiny, and it is, but when you're vaccinating hundreds of millions of people, those rare cases add up to real human suffering. In the United States, by the late 1990s, the only cases of paralytic polio were actually caused by the vaccine itself, because wild poliovirus had been eliminated from the country. So in 2000, The U.S. switched back to the inactivated Salk-type vaccine exclusively. It's a remarkable story. Two brilliant scientists, two different engineering solutions, each with genuine advantages and genuine risks, and the optimal choice shifting over time as the epidemiological situation changed. Now, both live attenuated and inactivated vaccines have been workhorses of public health for decades. Your measles, mumps, and rubella vaccine, the MMR, uses live attenuated viruses. The flu shot you get each fall is typically an inactivated vaccine, though there's also a live attenuated nasal spray version. The yellow fever vaccine, developed by Max Theiler in the 1930s, he won the Nobel Prize for it in 1951, is a live attenuated vaccine that's still used today, essentially unchanged. One of the most successful vaccines ever created. A single dose provides protection for decades, possibly for life. But there are viruses that stubbornly resist these traditional approaches. HIV is the most notorious example. Since the virus was identified in 1983, researchers have been trying to develop a vaccine, and the challenge is immense. HIV mutates at a ferocious rate, far faster than influenza. It attacks the very CD4+ helper T cells that coordinate the immune response, the generals of the army, if you remember that analogy. It integrates into the host genome, creating latent reservoirs that the immune system can't see. And its surface protein, the envelope glycoprotein, is heavily shielded by sugar molecules that make it difficult for antibodies to get a grip. Every major vaccine trial for HIV has failed or shown only modest results. The RV144 trial in Thailand, reported in 2009, showed about 31% efficacy, better than nothing, statistically significant, but not nearly enough for a practical vaccine. HIV taught the field humility, and it also pushed researchers toward entirely new approaches to vaccine design. And this is where the story gets really interesting, because the late 20th and early 21st centuries saw a revolution in how we think about vaccines. Instead of using whole pathogens, alive or dead, what if you could use just a piece of the pathogen, just the specific protein that the immune system needs to see? This is the subunit vaccine concept, and it's been around since the 1980s. The hepatitis B vaccine, for instance, uses just the surface antigen of the virus, produced by genetically engineered yeast cells. You take the gene for the hepatitis B surface protein, insert it into yeast DNA, let the yeast manufacture the protein, purify it, and inject it. No virus involved at all. It's elegant, it's safe, and it works beautifully. That vaccine has prevented millions of cases of liver cancer worldwide, because chronic hepatitis B infection is a major cause of hepatocellular carcinoma. But subunit vaccines sometimes don't provoke a strong enough immune response on their own. The immune system evolved to respond to things that look like actual threats. Whole pathogens covered in multiple danger signals. A single purified protein sitting in a syringe doesn't always set off the alarms loudly enough. So you need adjuvants, substances mixed in with the antigen that essentially wave a red flag and tell the immune system, hey, pay attention, this is important. Aluminum salts have been used as adjuvants since the 1920s, and honestly, for decades nobody fully understood why they worked. They just did. More recently, newer adjuvants have been developed that activate specific pattern recognition receptors, those same PRRs we talked about in innate immunity, to deliberately trigger the inflammatory signals that jumpstart the adaptive response. Then came the really radical ideas. What if, instead of injecting a protein, you injected the instructions for making the protein and let the body's own cells manufacture it? This is the concept behind nucleic acid vaccines, DNA vaccines, and RNA vaccines. The idea had been floating around since the 1990s. In 1990, researchers at the University of Wisconsin showed that injecting naked DNA into mouse muscle cells could lead to protein expression. The cells would take up the DNA, transcribe it into mRNA, translate it into protein, and the immune system would respond. It was a proof of concept, and it was exciting. But DNA vaccines faced practical hurdles. They were hard to get into cells efficiently, and there were theoretical concerns about integration into the host genome. RNA vaccines, specifically messenger RNA vaccines, took a different path, and the story of how they went from a fringe idea to the technology that helped end a pandemic is one of the great scientific narratives of our time. The key figure here is Katalin Karikó, a Hungarian-born biochemist who spent decades working on mRNA therapeutics at the University of Pennsylvania, largely in obscurity. For years, she couldn't get grants funded. She was demoted from the tenure track. The scientific establishment was skeptical. Injected mRNA provoked too much inflammation. It was unstable. It degraded too quickly. But Karikó kept working. And in the early 2000s, she and her colleague Drew Weissman made a crucial discovery. They found that by substituting one of the 4 nucleoside bases in the mRNA, replacing uridine with pseudouridine, they could dramatically reduce the inflammatory response while still allowing the mRNA to be translated into protein. That modification was the key that unlocked everything. It made mRNA therapeutically viable. Karikó and Weissman published their findings in 2005, and while the paper didn't make headlines at the time, it caught the attention of people who would later found BioNTech and Moderna. When SARS-CoV-2 emerged in late 2019, and the genetic sequence was published on January 11th, 2020. Those companies already had the platform ready. They didn't need to grow virus, didn't need to attenuate anything. didn't need to purify proteins from cell cultures. They just needed the sequence of the spike protein. That same spike protein that binds to ACE2, the one we talked about way back when we discussed viral entry. And they could synthesize the mRNA encoding it, wrap it in lipid nanoparticles to protect it and help it enter cells, and have a vaccine candidate ready for testing within days. Moderna designed their vaccine candidate in 2 days. 2 days. The first clinical batch was manufactured within about 6 weeks, and by December 2020, less than a year after the pandemic began, both the Pfizer-BioNTech and Moderna mRNA vaccines had received emergency use authorizations after large-scale trials, showing roughly 95% efficacy. Think about what that represents. From Jenner scraping cowpox pus off a milkmaid's hand, to a vaccine designed on a computer in a weekend, synthesized in a lab, and deployed globally within months. That arc of progress spans more than 2 centuries, and every step along the way, Pasteur's attenuated cultures, Salk's formaldehyde-killed virus, Sabin's oral drops, the recombinant hepatitis B protein, Carico's pseudouridine modification, each one built on everything that came before. And mRNA technology isn't just about COVID. Researchers are now developing mRNA vaccines for influenza, RSV, Zika, rabies, and even personalized cancer vaccines tailored to the specific mutations in an individual patient's tumor. Carico and Weissman won the Nobel Prize in Physiology or Medicine in 2023. And it's hard to think of a more deserved recognition. But vaccines, as powerful as they are, aren't the only tool we have against viruses. And for some viral infections, you need something that works after the virus has already gotten inside you and started replicating. You need antiviral drugs. And the story of how we learned to fight viruses with pharmaceuticals is its own fascinating chapter in the history of medicine. One that starts with a woman named Gertrude Elion and a deeply personal kind of grief. Gertrude Elion lost her grandfather to stomach cancer when she was 15 years old. It was 1933, and she watched him suffer, and she made a decision right then that she would devote her life to finding cures for disease. Now, that's the kind of origin story you hear sometimes, and it sounds almost too neat, too perfectly motivational. But by all accounts, that's really what happened. She enrolled at Hunter College in New York City, graduated with a chemistry degree in 1937, and then ran headfirst into the wall that women in science hit constantly in that era. She couldn't get a research position. Nobody wanted to hire a female chemist. She taught high school. She worked as a lab assistant. She took whatever she could get. And then World War II changed everything, the way it changed so many things, because suddenly men were overseas and laboratories needed people. Elion got a position at Burroughs Wellcome, a pharmaceutical company, working under a biochemist named George Hitchings. And the partnership between Elian and Hitchings would eventually transform how we design drugs. Here's what was different about their approach. Before Elian and Hitchings, most drug discovery was basically trial and error. You'd screen thousands of compounds, see if any of them happened to kill a pathogen or slow a disease. And if something worked, you often didn't fully understand why. Elian and Hitchings wanted to be more rational about it. They studied the biochemical differences between normal human cells and the things that invade them: cancer cells, bacteria, parasites, viruses. They focused on nucleic acid metabolism, on how cells build the building blocks of DNA and RNA, and their insight was beautifully simple in concept. If you could design a molecule that looked enough like a real nucleotide to fool a pathogen's enzymes, but was different enough to gum up the works, you could selectively disrupt the pathogen without destroying the patient's own cells. They called these molecules anti-metabolites, and the idea of rational drug design, starting from an understanding of biochemistry and working toward a molecule rather than just blindly screening, that was revolutionary. Elian developed drugs for leukemia, for gout, for organ transplant rejection, for herpes. That last one is where the antiviral story really picks up speed. In 1977, Elian and her team developed acyclovir, and it became one of the first truly effective antiviral drugs. Let's talk about how it works, because it's genuinely elegant. Acyclovir is a nucleoside analog. It looks like guanosine, one of the building blocks of DNA, but it's missing a critical piece of its sugar ring. When you take acyclovir, it gets into your cells, but here's the clever part. It doesn't do much in healthy cells. It needs to be activated first, converted into its active form by an enzyme called thymidine kinase. And human cells have a version of thymidine kinase, but it barely recognizes acyclovir. Herpes simplex virus, though, encodes its own thymidine kinase, and the viral version is much less picky. It grabs acyclovir and phosphorylates it, starts the activation process. Then normal cellular enzymes finish the job, converting it to acyclovir triphosphate. Now you've got this activated drug molecule concentrated specifically in infected cells, because only infected cells have the viral thymidine kinase to get the process started. And then acyclovir triphosphate competes with normal guanosine triphosphate for incorporation into the growing DNA chain by the viral DNA polymerase. The viral polymerase, like the viral thymidine kinase, is less discriminating than the human version. It's more likely to grab acyclovir and stick it into the DNA it's copying. But because acyclovir is missing that piece of its sugar ring, there's no place to attach the next nucleotide. The chain just stops. It's called chain termination, and it's lethal for viral replication. Think about the selectivity built into that mechanism. Two separate filters. The viral thymidine kinase activates the drug preferentially in infected cells, and the viral DNA polymerase incorporates it, preferentially into viral DNA. That's why acyclovir has relatively few side effects compared to earlier, cruder antiviral attempts. It's a sniper rifle, not a shotgun. Elion and Hitchings shared the Nobel Prize in Physiology or Medicine in 1988, and Elion, who never earned a PhD because she kept having to choose between work and graduate school in an era that didn't make room for women in science, became one of the most celebrated pharmacologists in history. Now, acyclovir works against herpes viruses because of that specific thymidine kinase trick. But not every virus gives you such a convenient target. When HIV emerged in the 1980s, the urgency to find antiviral drugs became desperate, and the approach had to be tailored to a completely different kind of virus. Remember, HIV is a retrovirus. It uses reverse transcriptase to convert its RNA genome into DNA and then integrase to stitch that DNA into the host chromosome. Those are viral enzymes that human cells don't have, which makes them beautiful drug targets. The first HIV drug approved was AZT, azidothiamidine, also called zidovudine, in 1987, and the story of AZT is complicated and bittersweet. The compound had actually been synthesized back in 1964 as a potential cancer drug, but it didn't work well against cancer and sat on a shelf for years. When researchers at the National Cancer Institute began screening compounds against HIV in the mid-1980s, AZT turned out to inhibit reverse transcriptase. It's another nucleoside analog like acyclovir, but targeting a different enzyme. Reverse transcriptase grabs AZT, incorporates it into the growing DNA chain, and the chain terminates because AZT has an azido group where the hydroxyl should be. No next nucleotide can attach. AZT was approved after a clinical trial that was stopped early because the difference between the treatment and placebo groups was so stark. Patients on AZT were surviving. Patients on placebo were dying. But here's the heartbreaking part. AZT alone wasn't enough. HIV mutates so rapidly, remember, because reversed transcriptase is sloppy. It makes mistakes, and those mistakes generate variants. Some of those variants have slightly altered reversed transcriptase that doesn't grab AZT as readily. Within months, resistance would develop, and the virus would come roaring back. For years in the late 1980s and early 1990s, an HIV diagnosis was still essentially a death sentence, just a slightly delayed one. Patients would go on AZT, improve for a while, and then decline as resistant virus took over. The breakthrough came from a concept that seems obvious in retrospect, but required real intellectual courage to push through. Combination therapy hit the virus with multiple drugs targeting different enzymes simultaneously. If the virus mutates to resist one drug, the other drugs are still working. For the virus to escape all of them at once, it would need to develop multiple specific mutations simultaneously, and the probability of that is vanishingly small. In 1996, researchers demonstrated that combining nucleoside analogues like AZT with a new class of drugs called protease inhibitors could suppress HIV to undetectable levels in the blood. Protease inhibitors target a different step entirely. Remember how HIV makes its proteins as one long polyprotein that needs to be cut into functional pieces by the viral protease enzyme? Block that protease, and the virus produces defective, immature, non-infectious particles. The combination approach became known as highly active antiretroviral therapy, or HAART, and it transformed HIV from a death sentence into a manageable chronic condition almost overnight. The 1996 International AIDS Conference in Vancouver is remembered as the moment everything changed. Researchers presented data showing viral loads dropping below detection limits, immune systems recovering, people getting up out of hospital beds and going back to their lives. And here's what's really worth sitting with for a moment. The drugs that made HAART possible came from understanding exactly how the virus works. Its reverse transcriptase, its protease, its integrase. Every one of those drug targets was identified through basic virology research, the kind of painstaking work we talked about earlier, people mapping out replication cycles and characterizing enzymes. The science of understanding viruses and the science of fighting them are the same science. Today, a person diagnosed with HIV who starts antiretroviral therapy and stays on it can expect a near-normal lifespan. There are now drugs targeting nearly every step of the HIV life cycle. Entry inhibitors that block the virus from fusing with the cell, integrase inhibitors that prevent the viral DNA from being stitched into the chromosome, and those combinations are so effective that people on successful treatment achieve what's called an undetectable viral load, meaning they can't transmit the virus to others. Undetectable equals untransmittable. That's not just a slogan. It's backed by large clinical studies involving thousands of couples. The same rational drug design philosophy has given us direct-acting antivirals for hepatitis C, and this is another one of those stories that doesn't get enough attention for how remarkable it is. Hepatitis C was identified in 1989, and for decades, the only treatment was interferon injections combined with ribavirin, a grueling regimen with terrible side effects and cure rates that were, depending on the viral genotype, sometimes below 50%. But researchers mapped the hepatitis C virus's replication machinery in exquisite detail, identified key enzymes, the NS3/4A protease, the NS5A protein, the NS5B RNA polymerase, and designed drugs to inhibit each of them. By 2014, combination pills like sofosbuvir paired with ledipasvir could cure hepatitis C in over 95% of patients with just 12 weeks of oral medication and minimal side effects. Cure. Not manage, not suppress. Cure. A viral infection that had been destroying livers and causing cancer for decades, cured with a bottle of pills. If that doesn't quietly astonish you, think about what it would have sounded like to a physician in 1990. But for all these triumphs, there are viruses we still can't cure and diseases we still can't fully prevent. HIV's latent reservoir, those integrated copies of viral DNA hiding silently in long-lived memory T cells, means that even though we can suppress the virus, we can't yet eliminate it from the body entirely. Stop the drugs, and the virus comes back. Researchers are working on shock and kill strategies to flush the virus out of hiding and then destroy it. But it's proven incredibly difficult. And for some viruses, we still don't have good antivirals at all. When a novel virus emerges, a new influenza strain, a new coronavirus, there's often a gap between the start of the outbreak and the availability of effective treatments. That gap is where the damage happens, and closing it faster is one of the great challenges of modern virology and pandemic preparedness. And it's worth sitting with that idea for a moment, because so much of what we've talked about tonight, all those decades of discovery, all those brilliant people chipping away at how viruses work, converges right here at the question of speed. How fast can we identify a new threat, understand it, and build something to fight it? Because if you rewind even 20 years, the answer was not fast enough, not nearly fast enough. The typical timeline for developing a vaccine used to be measured in decades, not months. You had to isolate the virus, grow it in culture, figure out how to weaken or inactivate it, test it in animals, test it in small groups of people, test it in larger groups, scale up manufacturing, get regulatory approval, and each of those steps could take years. The mumps vaccine, which held the speed record for a long time, took about 4 years from sample collection to licensure, and that was considered breathtakingly fast. 4 years. And for many diseases, the timeline stretched to 10, 15, 20 years or more. Some vaccines never arrived at all, so when a new virus appeared, SARS in 2002, H1N1 influenza in 2009, MERS in 2012, Ebola in 2014, Zika in 2015. The world was essentially running on borrowed time, relying on containment, quarantine, supportive care, and hoping the outbreak burned itself out before a vaccine or antiviral could be ready. Sometimes that worked. SARS was contained through aggressive public health measures before a vaccine was needed, but the underlying vulnerability was always there, like a crack in a dam that hadn't been tested by a truly massive flood. And then, of course, came the flood. In late December 2019, clinicians in Wuhan, China, began noticing clusters of pneumonia cases with no clear cause. By January 10th, 2020, Chinese scientists had sequenced the genome of the responsible pathogen, a novel coronavirus soon named SARS-CoV-2, and posted it publicly. And here's where everything we've been talking about tonight comes together in a way that would have seemed like science fiction to someone like Wendell Stanley or even Jonas Salk. Within days of that sequence being published, researchers at Moderna in Cambridge, Massachusetts, and at biotech in Mainz, Germany, had already designed candidate mRNA vaccines, not weeks, days, because the platform was ready. Remember Katalin Karikó and Drew Weissman's work on modified nucleosides, the pseudouridine substitution that made synthetic mRNA tolerable to the immune system without triggering destructive inflammation. That foundational work, done years earlier when almost nobody was paying attention, meant that the technology was sitting there waiting. All you needed was the genetic sequence of the spike protein, and you could plug it into the mRNA template like swapping a file on a computer. The manufacturing process didn't change. The lipid nanoparticle delivery system didn't change. Only the instructions changed. Moderna finalized its vaccine sequence on January 13th, 2020, just 2 days after the viral genome was shared. 2 days. They shipped the first batch of vaccine to the National Institutes of Health for Phase 1 clinical trials by February 24th. The first human received a dose on March 16th, barely 2 months after the virus had been genetically identified, and Pfizer-BioNTech was on a parallel track, moving with similar speed. Think about what that means in the context of everything we've discussed, from Ivanovsky's mysterious filterable agent in 1892, from Bajerink's Contagium vivum fluidum, from Stanley's crystallized tobacco mosaic virus, over a century of accumulated understanding about what viruses are, how they replicate, how our immune systems recognize them, how to present antigens safely. All of that knowledge compressed into a response time of weeks. Now, the clinical trials still took months, and rightly so. You can't skip safety testing, and the Phase 3 trials that enrolled tens of thousands of volunteers were essential for establishing both safety and efficacy. But even those trials were designed and executed with unprecedented speed, partly because the pandemic itself provided, tragically, no shortage of viral exposure in the placebo groups. By November 2020, both Moderna and Pfizer-BioNTech reported efficacy results around 95%, numbers that stunned even optimistic vaccinologists. By December, emergency use authorizations were granted. Less than a year from genome sequence to authorized vaccine, that old record of 4 years for the mumps vaccine wasn't just broken, it was shattered. And here's something worth reflecting on quietly. The speed wasn't the result of cutting corners. It was the result of decades of unglamorous foundational work finally paying off. Carrico spent years struggling to get funding, getting demoted, watching her grant applications get rejected, all while pursuing the basic science of mRNA modification. Weissman was studying dendritic cells, those same antigen-presenting cells we talked about earlier, the ones that pick up foreign material and show it to T cells. And their collaboration was driven by pure curiosity about how immune cells respond to different forms of RNA. They weren't trying to build a pandemic vaccine. They were trying to understand biology, and that understanding, years later, saved millions of lives. But mRNA vaccines aren't the only technology that emerged from this convergence of knowledge. Let's talk about something else that happened during the pandemic, because it illustrates a different dimension of how our understanding of viruses has matured. When SARS-CoV-2 began spreading, researchers needed diagnostic tests immediately, and they got them thanks to a technique called polymerase chain reaction, PCR, which you've almost certainly heard of by now. PCR was invented by Kary Mullis in 1983, and it allows you to take a tiny sample of genetic material and amplify it millions of times until there's enough to detect. For virus detection, you combine PCR with reverse transcription because SARS-CoV-2 is an RNA virus. You first convert its RNA into DNA using reversed transcriptase, that same enzyme Howard Temin and David Baltimore discovered in 1970. So the diagnostic test that became the global standard for COVID detection relies directly on an enzyme discovered over 50 years ago by researchers studying RNA tumor viruses in chickens. The connections are everywhere once you start looking, and the genomic surveillance, the ability to track how the virus was mutating in real time to identify new variants like Alpha, Delta, Omicron, that depended on rapid, cheap genome sequencing technology that simply didn't exist a generation ago. Scientists around the world were sequencing viral genomes from patient samples and uploading them to shared databases within hours, creating a kind of living map of viral evolution. Remember when we talked about antigenic drift, those small, gradual mutations that change the shape of surface proteins? That's exactly what was being tracked. Each new variant represented a slightly different configuration of the spike protein, and researchers could see those changes accumulating in real time, predict which mutations might affect transmissibility or immune evasion, and begin updating vaccines accordingly. This is where the story comes full circle in a way that feels almost poetic. The same basic principles that Gruber and von Purkett were puzzling over more than a century ago. How does the body recognize something it's seen before? And how does that recognition change when the invader changes? Are the exact questions that drove the development of updated booster shots targeting Omicron subvariants. The science got more precise, the tools got more powerful, but the fundamental questions haven't changed. Now, I want to be honest with you about something, because this wouldn't be a fair telling of the story if I only talked about the triumphs. The pandemic also exposed enormous gaps in global vaccine distribution, in public trust, in our ability to coordinate a coherent response across borders and political systems. Vaccines that were available in wealthy countries by early 2021 didn't reach many lower-income countries until much later, and by then the virus had continued to circulate and mutate. The biology was a success story. The logistics and equity were, in many ways, a failure. And that matters because a virus doesn't care about national borders. As long as it's replicating somewhere, it's evolving somewhere. And evolution is the engine that drives new variants. There's also the sobering reality that we were, in some sense, lucky with SARS-CoV-2. Its fatality rate, while devastating in absolute numbers, millions of deaths worldwide, was far lower than what we'd see with a highly pathogenic avian influenza strain or a more lethal coronavirus. The next pandemic pathogen might not give us as much time, and that's why the work happening right now in labs and institutions around the world is focused on being even faster, even more prepared. Researchers are developing what they call prototype pathogen approaches, pre-designing vaccine candidates for entire families of viruses, so that when a new member of that family emerges, the response time shrinks from months to weeks or even days. There are efforts to build universal influenza vaccines that target the conserved stalk region of the hemagglutinin protein, rather than the highly variable head going after the part of the virus that doesn't change much. So the vaccine works against many strains at once. Similar strategies are being explored for coronaviruses, aiming for pan-coronavirus vaccines that could protect against not just known viruses, but future ones that haven't spilled over from animal reservoirs yet. And the antiviral side is advancing too. During the pandemic, two notable oral antivirals emerged: molnupiravir, which works by introducing errors into the viral RNA during replication, essentially causing the virus to mutate itself to death, and nirmatrelvir, the active component of Paxlovid, which inhibits the SARS-CoV-2 main protease, an enzyme the virus needs to process its polyproteins into functional pieces. That protease inhibitor approach is directly descended from the same rational drug design philosophy that Gertrude Elion and George Hitchings pioneered decades ago. You understand the enzyme, you understand its structure, and you build a molecule that fits into its active site and jams it. Same logic, new target. So where does all of this leave us? You've been lying here, maybe still awake, maybe drifting, while we've wandered through over a century of discovery. From Dmitry Ivanovsky filtering tobacco leaf extract through porcelain in 1892 and finding that something impossibly small could still cause disease. From Martinus Beijerinck daring to propose that the infectious agent wasn't a bacterium at all, but something new, something liquid and living, in a way no one had imagined, from Wendell Stanley crystallizing a virus and blurring the line between chemistry and life, from Rosalind Franklin and Aaron Klug revealing the geometric elegance of viral capsids, from Howard Temin stubbornly insisting that information could flow from RNA to DNA, until he and David Baltimore proved it and changed everything we thought we knew about genetics, from the quiet heroism of the Salk and Sabin vaccine campaigns and the astonishing global effort that wiped smallpox from the face of the earth, from Gertrude Elion, who wasn't allowed to pursue a PhD because of her gender, going on to develop drugs that saved countless lives and winning a Nobel Prize, from Catalin Carrico, demoted and defunded, persisting with mRNA research that became the backbone of the fastest vaccine development in history. What strikes me most when you step back and look at the whole arc is how much of this story is about patience. Viruses are patient. They've been evolving for billions of years, long before anything we'd recognize as human walked the earth. And the people who figured them out were patient too, in their own way. They asked questions that took decades to answer. They built on each other's work across generations and continents, often without knowing where it would lead. The person who discovered reverse transcriptase wasn't thinking about HIV. HIV hadn't been identified yet. The person who figured out how to modify mRNA wasn't thinking about a pandemic that was still 15 years away. They were just trying to understand something, and that understanding, accumulated slowly, piece by piece, turned out to be exactly what the world needed when the moment came. Viruses are still out there tonight, doing what they've always done, drifting on air currents, hitching rides in droplets of water, slipping between cells, copying themselves with that strange, relentless efficiency. They're in the soil, in the ocean, in the animals sleeping in forests you'll never visit. Most of them will never affect you at all, but they're part of the fabric of life on this planet in ways we're only beginning to appreciate, shaping ecosystems, driving evolution, even contributing genes that became essential parts of our own biology. Remember syncytin, that protein in the human placenta that came from an ancient retrovirus. You're here, in part, because a virus infected one of your distant ancestors and left behind a gift. So as you settle deeper into wherever you are right now, your pillow, your blanket, the quiet of this late hour, maybe carry that thought with you into sleep, that the boundary between us and viruses isn't as sharp as it seems, that the story of how we learned to understand them is really a story about curiosity, about persistence, about people who refused to stop asking why. And that story isn't over. It's still being written in labs and hospitals and field stations around the world by people you'll never meet, working on problems that might not matter for years or might matter tomorrow, but they're working. And that, I think, is a good thought to fall asleep to. Good night.